What drives us at CIRA is improving the quality of life of the people affected by vision loss. So CIRA has the ability to look at all of the major diseases that cause vision loss and blindness. That's unique in Australia.
At CIRA we have a very large research program into macular disease. We cover really the basic science area, so trying to understand what causes AMD, all the way through to clinical trials.
Inherited retinal disease is the most common cause of blindness in working-aged Australians, and so we're very interested in learning more about those conditions and developing some new treatments.
There are many people suffering from glaucoma worldwide and in Australia.
My job is to diagnose glaucoma earlier so that we can prevent vision loss in people, and we can actually tailor treatments for them instead of just using the one treatment to treat everyone.
Our research is trying to understand the communication between the cells that are located in the retina, and we are investigating these cells, how they talk to the blood vessels.
If we understand this, we can work on getting new therapies out there.
Diabetes is becoming more and more common in Australia. 1 in 5 of those people actually develop diabetic eye disease. A lot of the treatments that we have are for late stages where you've already lost vision, and so what we really need to look at is being better at detecting early eye disease.
We've had incredible advances in our ability to image the eye in incredible levels of detail.
We're seeing things that we could not see through any other imaging method available currently in the clinic. Changes in the brain and in the eye for Alzheimer's disease develop up to 20 years before the onset of memory impairment.
Genomics is changing the way that we diagnose, treat and understand different eye diseases.
Gene therapy has been a game changer for eye disease. For most of my career, I've been telling patients with inherited retinal diseases that there's absolutely nothing we can do to improve their function. And that is now starting to change.
My lab focuses on using stem cells, so we are developing a gene therapy to regenerate the photoreceptor to restore vision in the patient.
Currently, young patients living with Usher syndrome, there's not any cure. So with the latest genetic technology, my team is developing gene therapy that can be fundamental, cure this disease.
Tissue for Research helps provide understanding to our researchers here at CIRA and elsewhere about the eye.
And the idea is that we will develop a tissue-engineered cornea. This will mean that we won't need to use donors for every single case. We'll be able to use one donor and amplify that to 50 different corneas.
I'm really excited about the future of CIRA.
It's a really exciting time. We can see a big pipeline of discovery.
Talented researchers with new ways of doing things.
New ideas, new innovations, new medicines, new therapies and devices coming from around the world.
We're right at the cusp of that big transformative step forward.
Centre for Eye Research Australia, Cerulean Clinical Trials, Lions Eye Donation Service, Melbourne. Hope in Sight, cira.org.au. Donate today.
Well, good evening, everyone. I'm Keith Martin. I'm Managing Director of the Centre for Eye Research Australia and Ringland Anderson Professor of Ophthalmology at the University of Melbourne. And I'm also the Chair of Cerulean Clinical Trials, which is CIRA's new not-for-profit clinical trials centre. So I'd like to start by acknowledging the traditional custodians of the land where the Melbourne Museum sits. That's the Wurundjeri and the Bunurong people of the Kulin Nation. I pay my respects to their elders, past and present, and to their families. And I extend my respects to any Aboriginal or Torres Strait Islander peoples who are here today. And I think it's really important to remember that First Nations peoples they continue to be disproportionately impacted by vision loss and blindness. And that's something that the eye research community and the eye health community are really trying to address through research and improvements in clinical services. I'd also like to highlight the effort of many of our researchers and clinical colleagues to close the Indigenous eye health gap. Now that includes friends who are here tonight, and we've got people and representatives from the Fred Hollows Foundation, from Vision 2020 Australia, from Vision Australia, from the University of Melbourne, and from the Royal Victorian Eye and Ear Hospital. I'd also like to acknowledge CIRA's founder, Melbourne Laureate Professor Emeritus Hugh Taylor, AC, who established the University of Melbourne's Indigenous High Unit and established CIRA in 1996 also. So, before we begin, just a few housekeeping matters. The bathrooms are out the back and to the right. In the unlikely event of emergency, don't run for your lives. Follow the instructions of the staff and evacuate to the main foyer entrance, and you'll be guided out to Rathdown Street. The other thing that I'd like to say is that it's a great honour to have so many of you here to honour Gerald Crock for the 2024 Crock Lecture. In terms of other welcomes, I'd like to welcome Victoria's Chief Scientist, Dr. Amanda Caples. We have Barinder Giddy here, who's the Chief Medical Officer of the Royal Victoria and Eye and Ear Hospital, and other members of the executive team here too. We've got hospital board directors, so Susan Stradinis is online, and Bruce Ryan is here representing the board. We have colleagues from the University of Melbourne, led by Associate Professor Andrew Mehta from the Department of Optometry and Vision Sciences. And we also have CIRA board and CIRA Foundation members. So Simon Bruin is a director on both the hospital and CIRA board. Professor Andrew Cuthbertson, AO, is Deputy Chancellor of the University of Melbourne, is also a CIRA board member and joining us online. Suwani Dharmalingam and Christine Edwards and Darren Kelly, who are also board members of our CIRA board, and Darren is also on the clinical trials board, and also Serge Scrofani is here also today. So we have also our new CEO of Cerulia, Michelle Gallagher, and our new Cerulia board director, Nick Northcott, who are all very welcome. And also representatives of our consumer advisory group, Jane Cherry and Colleen Lewis. And a big thank you to Alex Grimwade, who's chair of CIRA's Philanthropic Committee, does so much to support our research together with Fred Grimwade. And thanks to all our philanthropic supporters also in the audience, and our dedicated staff and student, and most importantly to members of the Crock family who are here today. And there are a few health issues that have prevented some of the other members of the Crock family being here today, but we're really grateful for those that have come tonight. So for those of you new to the CIRA community, I'll just share a little piece of history. So it was 1963 when Gerald Crock was appointed to the University of Melbourne as the first Ringland Anderson Professor of Ophthalmology, and that was at the very young age of 34, which is just incredible to think that he took the role at that age. And his appointment was the first medical specialty chair of any discipline in Australia, and he was only the second chair of ophthalmology in the British Commonwealth at the time. And he established the university's Department of Ophthalmology and much of the Royal Victorian Eye and Ear Hospital as it is today. And he had a real drive to discover new and better ways to save sight, and that pioneering spirit has left a large legacy and remains an inspiration for many of us through to this day. So each year the Crock Lecture celebrates innovation in eye research, and I was honoured to give this lecture last year. And during that lecture I shared a video of a child with a rare genetic disease who has received a gene therapy called Luxturna, which you'll hear a little bit more about tonight. So I thought it was a good opportunity just to remind us what we're trying to achieve and what has been achieved with one of the children who was treated with this treatment last year.
Orange? Yeah, you're okay. Here is purple. Yes, here is red.
So children with this disease have very poor vision in low light conditions.
Here is blue.
And so this little boy had real trouble navigating around. You see, he's feeling his way around with his feet.
Bumpy. Keep going. Bumpy, bumpy, bumpy. You got it.
And he's not even looking down because it doesn't really help him very much in these light levels to look down. So really struggling. And you may have heard he said at the start, "I'm scared of the dark," was his comment at the beginning. 3 weeks after the gene therapy that was delivered in the Eye and Ear Hospital, this is the same child in the same light level. And he said there, "I'm the winner," if we had the sound playing. So and he is certainly the winner in that. So, and this is the sort of thing that we really want to be able to do more. And as I said last year, this is really, truly life-changing treatment. And so tonight we're thrilled to be able to introduce to our CIRA community the co-creator of that medicine, which was the first gene therapy for any inherited disease to come to market. That's Professor Jean Bennett. And Jean is Emeritus Professor of Ophthalmology at the Perelman School of Medicine at the University of Pennsylvania. And she's an inspiration to scientists around the world through this breakthrough of bringing this first gene therapy for an inherited retinal disease from the bench all the way through to the clinic, all the way through to transforming lives in Melbourne and Victoria. And that achievement has really paved the way for the cutting-edge research that's now underway at CIRA on the next generation of cures for genetic blindness. And that includes the work of my colleague at CIRA and at the Eye and Ear Hospital, Dr. Tom Edwards. So Tom is the co-lead of our retinal gene therapy unit, which is conducting research to develop gene therapies to prevent blindness from genetic eye diseases. And he works along with Associate Professor Lauren Ayton, who lead what we call the Venture Study, which is a registry of more than 450 people living with inherited retinal disease that aims to build our understanding of these diseases and match patients to clinical trials in the future. So this is no longer just classifying these diseases, this is really aimed at matching them to new treatments, which I think is very exciting. So Tom was also an ophthalmologist and a vitreoretinal surgeon, and in 2014 he was awarded a fellowship to study at Oxford University under Professor Robert McLaren, where he worked on a world-first clinical trial of gene therapy to treat another inherited retinal disease called choroideremia. So he brought those skills back to Australia, and in addition to his work at CIRA, he's a vitreoretinal surgeon at the Eye and Ear Hospital. And he's one of only a handful of surgeons in Australia who have delivered Luxturna to patients. So please welcome Tom to introduce our special guest tonight.
We can take that slide down. Thanks, Keith. Well, as an ophthalmologist, one of the most challenging aspects of my job is to tell a patient or their parents that they may have an inherited retinal disease and that there is no treatment for their condition. Telling a young person on the cusp of adulthood and independence that they may never have a driver's license, for example, something so many of us take for granted, is one of the hardest things for a young patient to hear, and one I dread. It's a conversation I dread having. It's also an incredibly sensitive conversation when discussing the risk of experiencing profound vision loss and the impact it could have on careers, family, and other loved ones. Now, a lot of people have never heard of inherited retinal diseases, but it's actually the leading cause of blindness in working-aged people, estimated to affect up to 17,000 Australians. And until very recently, inherited retinal disease, as Keith said, was considered incurable, and that nothing could be done to save the sight of someone with such a condition. But, as we'll hear tonight, that is changing. In the past decade, rapid advances in technology and groundbreaking science led by Professor Jean Bennett have been converting hope into therapeutic reality for some inherited retinal diseases. In her role at the University of Pennsylvania, Jean has spent 30 years developing gene-based strategies for treating inherited retinal diseases. Jean was the scientific director of clinical studies at the Children's Hospital of Philadelphia, leading pivotal work that in 2017 resulted in the first FDA-approved gene therapy for an inherited retinal disease. And that treatment was for a form of blindness caused by a gene called RPE65, also known as Leber's congenital amaurosis. In 2023, Jean became Professor Emeritus, but continues to develop gene therapies for blinding diseases at UPenn and with other biotechnology and academic groups. She's received many awards, some of which I will list here. The Smithsonian Ingenuity Award, the Champalimaud Award, the Sanford Lorraine Cross Award, the Harrington Prize, and the Helen Keller Prize in Vision Research. She's a member of the National Academy of Medicine, National Academy of Science, the Association of American Physicians, and the American Academy of Arts and Sciences. And I just found out tonight she used to be, and perhaps still is, a very keen diver. Her research has given eye doctors like me the opportunity to deliver a life-changing treatment to patients, as you've just seen there in Keith's wonderful video. And it's Jean who's been such an inspiring role model to us all, and a trailblazing scientist who's had a far-reaching impact that has stretched all the way to our patients treated with Luxturna here in Melbourne. Our team feels very fortunate, therefore, to have participated in the global impact of Jean's bench-to-bedside research. And so it's my enormous privilege to introduce to you this evening Professor Jean Bennett, FM Kirby Professor of Ophthalmology at the Perelman School of Medicine, University of Pennsylvania, to deliver the 15th Annual Gerard Kroc Lecture. Jean.
What a warm, wonderful introduction. Thank you so much, Keith and Tom, and everybody I've met today and on my visit to Melbourne. This is extraordinary. And a special reach out to the Kroc family. It's just such an honor to be asked to give this lecture in honor of this incredible man whose work I remember from my time at Johns Hopkins. I believe he's had a stint at Wilmer Eye Institute there, where his ophthalmoscope is famous. And so thank you all. It's been an extraordinary visit. So before I get going, I'd like to acknowledge the traditional custodians of the land and their elders, past, present, and emerging. And I want you all to think about what it would be like to live a life of darkness. Studies have shown that people fear blindness more than death. And studies have also showed that people fear blindness second only to cancer. Blindness affects many, many people. You've heard about the numbers in Australia. In the United States, there are some 7 million people blind due to various inherited forms of retinal and macular degeneration. And it's been it affects their careers, what they can do independently, and so forth. But as you've heard, there is hope, and the hope is personified by, first of all, this dog here who was born blind due to a spontaneous mutation in the RP65 gene, but went through one shot of gene therapy and could see. He's at his third visit to Congress talking about the importance of biomedical research. And the first 8-year-old boy in our study who came to the hospital walking with his blind cane and holding his parents' hands in the hospital. And then a few weeks later, he's doing things which are clearly visual, including playing sports, riding his bicycle to school, seeing what the teacher is writing on the whiteboard, and playing video games. Maybe some things his parents would rather him not be doing, but living the life of a normal 9-year-old boy. So I'm going to tell you the story about how this was developed. First, I want to acknowledge my potential conflicts of interest. I am a co-author of the patent that led to this, but my husband and I, who's also a co-author, waived any potential financial gain so that we could work on these studies without any conflict. And I also want to acknowledge that it took a village to carry out these studies, and I've listed all of these people here who were really critical to various aspects of the study. So where did this all start for me? It started a long time ago, in about 1985. I knew when I went to medical school that I wanted to learn about the diseases so that I could develop gene therapy. There had been enormous progress in developing the ability to manipulate genes, and I was really especially excited by a study which showed that you could put a gene for growth hormone in a mouse and turn it into a gigantic mouse. And that, of course, inspired a lot of jokes about, well, you need better mouse traps, and this mouse is going to knock on your door and ask for a cheeseburger, et cetera. But of course, what I wanted to do was figure out how we could use gene delivery as an intervention. And there, fast forward to medical school, which is some 40 years ago, before cell phones, but when apps were applications to jobs, when there was no Google, there were only a few television stations, et cetera. My husband said to me after our first successful gene transfer experiment, which was the birth of our daughter, Sarah, do you think we could transfer genes to the retina? And he had been working with a person who was one of the first people to characterize the genetic basis of different forms of retinal disease. And in the glow of our new daughter, I said, yes. But what I didn't tell him was the problems. Well, first of all, what is gene therapy? Techniques that harness DNA or RNA to treat or prevent disease. So any product that can be encoded by DNA or RNA is gene therapy. This is impossible to do one by one in our cells. I had been injecting single cells, and here eggs, nuclei of eggs with DNA in my laboratory studies. That would be impossible to do in the billions of cells in our body. But viruses can deliver genes very, very efficiently. And so for a chronic disease, you need long-term expression of the gene in the appropriate cells. And this shows the first two approved gene therapies, one of which you've heard the name already, Luxturna. But anyway, when we started, there were no genes identified that cause blindness. There were no animal models, no natural history of the diseases, no vectors, i.e. reagents that help deliver DNA, which is highly charged and really difficult to get into cells without some tricks. No surgical methods or ways of measuring the effect of your treatment, outcome measures, and certainly no funding. And the field has grown enormously since then, thanks to patient advocates. And in our country, Gordon and Luli Gund, who started Foundation Fighting Blindness, and in this country, Retina International, and all over the world, it's patients who really kind of fueled the research and made it grow, helped us identify the genes. And they also provided the initial resources to get going to tackle all these problems. But honestly, if I had known how many problems there were, I don't know if I'd go forward. We were naive, and fortunately. But one of the first questions was how to figure out how to deliver genes to the retinal cells. And we started with non-viral methods, which work, but only for a very small amount of time, a couple weeks. And it tested various recombinant viruses, which are neutered to prevent the multiplication of these viruses and cause the harmful symptoms that they could cause in the wild. Basically to use them to piggyback genes into the cells of interest. And it started out, people were really scared of this. We had to do this in biohazard level 4 facilities surrounded by really terrible viruses. But we survived that. And we found, we and others found that adeno-associated virus, which is a virus which is often a co-infection of the common cold virus, but has never been shown to cause a disease in humans or animals in the wild, was a great vector, a great way to deliver genes to the cells in the retina and other cells that don't divide. And you can see in that, on the right there, individual photoreceptors, which are expressing this green fluorescent protein derived from bioluminescent jellyfish 2 years after this monkey retina was injected. So it's long-lasting, durable, which is also something you want. So when we started working on RP65, we generated what's called a construct. It's the gene with an on switch, which is supercharged. It drives high levels of this expression, packed it into this virus shell, and my husband, Albert McGuire, who's a retina surgeon, figured out a way of delivering this, which is diagrammed in this figure on the right. It basically injects a blister. It separates 2 layers of the retina, which are normally tightly opposed, and delivers the gene so that it's exposed only to the cells in the retina that need fixing. And there were lots of other tricks. We figured out how to purify the vector, make sure that it was free of empty particles so that we weren't just delivering duds, and how to make sure that it doesn't stick to the inner surfaces of the plastic reagents which you use, like syringes and so forth. And then shown here is the surgery. You can see this very tiny needle, which is about the width of an eyelash, which delivers this reagent to the retina, causing this blister-like retinal detachment, exposing the cells in the retina which need the fixing. And amazingly, this flattens down within a day after the injection, leaving very little evidence that anything has happened in that retina. And so that is the technique which Tom has also used here to treat the patient that you saw. And he's had much experience with that, with multiple clinical trials. Anyway, going back to the dogs, shown here is one of the dogs who, before injection, he walks around, he bumps into objects because he can't see them, and then after an injection, he walks around avoiding the objects, wagging his tail, he's a lot happier. And then you can see down below, when a light illuminates the eye that is not injected, the pupil does not constrict the way it normally does if you see light, but the eye that has been injected has a brisk pupillary reflex, which is a really great objective measure whether this is working. And the dogs could read afterwards. Not really. But this is Lancelot, who was made Mutant of the Month by Nature Genetics some years ago. So what is this disease? It's a rare autosomal recessive disease classified as Leber's congenital amaurosis or early onset retinitis pigmentosa. And children are born with pretty severe vision. They can see light, but whatever vision they have is really, really poor, as you saw in that early film. But worse than that is it's slowly progressive, so that the cells in the retina degenerate. So after a couple of decades, they can hardly see anything at all. And often it's accompanied by nystagmus, which is abnormal rotatory movements of the eyes. You can see both in this little boy and also in the dog who's affected. And for us, it seemed like a really straightforward gene therapy target because the RP65 gene, which is mutated in both the dog and the human, it's an enzyme which helps produce a vitamin A derivative, which is essential for those photoreceptors to be able to see light. You probably were told by your parents, eat your carrots because that vitamin A moiety is necessary. Well, it's true. But here, these people cannot make that vitamin A moiety. And so if we deliver that enzyme, the idea was that we could correct it. But there were so many obstacles. We'd figured out how to do it, we figured out the doses and so forth, and we were about ready to start. And then there was a very hostile environment for gene therapy. There was the first gene therapy death in another trial we were not involved with, using a different vector, using really high dose delivery directly to the liver. But there had been a lot of hype about this, and people were totally, rightfully alarmed. And so there were reviews of this. All the gene therapy trials halted. All the funding decreased. It was blocked, and we were stuck. So it took some time to get back going, but it did get going. But there were numerous other obstacles wherever we turned. And I'll just show you some of these. First of all, there was no path, and you have to go through various regulatory bodies to get approval to try an experimental treatment, particularly if you want to try it in children. Children were our ultimate goal because, as I told you, it's a degenerative disease. And for gene therapy, you need the cells to be there to treat. If they're all gone, there's nothing left to treat. It could just cause harm. So we had to figure out the path, which involved numerous paperwork inquiry. And at that point in time, there was a committee called the Recombinant DNA Advisory Committee, the RAC, aptly named, which grilled us for 2 days. And right as we thought we were going to get full approvals, an ethicist got up and said, "My uncle was blind, and he was a millionaire, and he knew how to navigate the streets of New York City. It's okay to be blind." And we thought we lost it, but here's where the patient advocates came to the rescue. 2 parents of children with this disease got up and spoke about how difficult it is to raise a blind child, and how the parents get divorced usually, how much money it costs, how reading one book, a Harry Potter book in Braille, it came in 12 different cases. Bulkiness of materials, it took 12 people to get their child through one school day in the public school. And then they all voted unanimously to let us go forward. And these parents are actually parents of a child who has been treated in a different clinical trial, which Jim Ackland, who is in this audience, helped pave the way for in Philadelphia. And I spoke about that earlier this morning. And so the other thing is gene therapy trials at that time did not include children, and we had argued about the importance of including children, and now all those trials can include children as well. So another challenge was that all of our colleagues thought we were crazy. There were some funny comments like, it will never work, and I have no idea what they're talking about, but it sounds smart. And one of my favorites is from my friend Katie Carico, who has recently won the Nobel Prize for mRNA, saying, how about RNA therapy? And I think, yeah, that's RNA therapy is actually now being tested in gene therapy clinical trials. So we passed all those obstacles and we were ready to enroll, but we couldn't find any patients because nobody in the US had been genotyping and phenotyping patients. So our Italian colleagues had been way ahead of us. They had genotyped and phenotyped their patients, and they had a number of them who fit all of our inclusion criteria. And our first 4 subjects came from Naples, Italy. And so they saved the day for that. And these are some of the other concerns. When we consented people, since this was really the first retinal gene therapy trial, we didn't know what could go wrong. We'd done all of the safety studies we could and more than was required. In fact, my gold standard was, if my kids had this disease, would I allow them to participate? And the answer was yes. But we didn't know for sure if there could be cancer or death, or what if, in one of the questions, and these are all in the consent, what if you do not like your vision? Well, there are ways of removing your vision. And also, it was pretty amusing telling 8-year-old boys that they could not father a child for the next year. So they had their gene therapy, and this is what the subjects said. And I won't read this whole list, but one of the children said, I didn't know that my mom had wrinkles. Her mom didn't like that so much. But the point is that they all gained independence. They could go to school, they could go to restaurants and find their fork and plate and so forth, see stars. One of them asked us if he could have a baby now, and he was serious about that. He ended up having 7 children. None of them had carried the gene therapy vector, which showed that there was no what's called vertical transmission, which is good. But they all said, when can I have my second eye injected? Because they really liked the benefit from the first eye. So we had to go back to the drawing board, to our precious animals. What we did is do sequential administration, first eye, second eye, very high dose. The concern was there'd be an immune response and it would ruin the benefit in the first eye and prevent any benefit in the second. But you can see here, Venus is doing beautifully through this, running through this obstacle course, and as did her children. And shown here are Venus and Mercury. After they finished these experiments, we adopted them and they lived out their lives. In fact, they chased squirrels, which you probably don't have around here, which was a really good test of their vision, that they could see these creatures. I'm sure if they were in Australia, they'd be chasing creatures like this. We went slowly when we injected the contralateral eye of humans, because humans don't always follow the data that you see in animals. And these individuals, the first ones who were from Italy, stayed with us because they didn't have relatives who could stay and come to the clinic week after week. Their relatives had to work. And that was amazing, just to have one of them come downstairs and look in the mirror and say, Mamma mia, I see my face. Or I look out the window and see branches on trees and try to figure out what they were seeing, understand what their eyes were telling them. And in fact, that led to a lot of additional studies by my colleague Manzar Ashtari, looking at the effects on the neural pathways, the visual pathways. And in fact, it looks like the gene therapy actually strengthened, myelinated the pathways from the retina to the vision parts of the brain. One of the questions we had to deal with was how to demonstrate a beneficial effect. And we went to the FDA early on saying we wanted to use pupillometry, and I'd show you that pupillary light reflex. And they said, no, you can't use that. People don't care about their pupillary light reflex. You have to use something that's clinically meaningful. And they didn't define what clinically meaningful was, but at that time, the only approved outcome measure was reading the eye chart, which of course is clinically meaningful. But what do you do if you cannot even see an eye chart and you can't read the letters there? How do you measure a benefit? And so we were stunned for a while, but we put our heads together and came up with a functional vision course, reasoning that if somebody can walk around accurately and quickly, avoiding objects and not tripping over them, that could be clinically meaningful. And we had tested a little course prior to this. We thought we had seen that this could be a really effective measure. This is called the multi-luminance mobility test. So we developed this test and perfected it and went through different light levels and so forth, and reconfigured the test for each various ones of the tests given for the day, so they wouldn't measure these the pathways, because blind people are really good at doing that. And this shows an example of what we saw before and after. The left is at baseline, where the person just steps forward and immediately goes off the path, knocks over obstacles, and continues to go off path. You can see 2 years later, this person walks through this path perfectly, avoiding all the obstacles, stepping over the things in the way of the path, not using their feet to kick the way blind individuals do, and finding the path. And this outcome measure turned out to be really effective and has been used in some other trials. So the drug was approved in 2017, and there was a race in the United States for different centers to be the first to give this prescribed drug. Shown on that left is a little girl, 4-year-old girl at UCLA, whose mother took her out for Halloween a few weeks after her gene therapy, and she could not have been less interested in the candy. What she was so excited about was that she could see in the dark and run around in the street and avoid objects, which she hadn't been able to do before. And the other children at the other centers, one was really proud that he could achieve his lifelong dream of throwing away his blind cane. Another was more cerebral about how he could see the whiteboard and he could ride his bike really fast. But it was very exciting for us. And we are so thrilled that Australia has been treating patients with Luxturna, and I was really especially touched yesterday to meet one of the Luxturna recipients yesterday, just very cute little boy whose face I've covered here just for confidentiality. But this is just, I never get tired of hearing the stories and the amazing people who have helped bring this, particularly the patients, bring this to approval. So what is the current status of gene therapies? There are now 38 different gene therapy products approved by the US FDA, and 15 of those target genetic disease and are administered directly to the body. There are several different viruses that have now been approved. There are different adeno-associated viruses, the one we used, some herpes simplex viruses, and one adenovirus. And there are a lot of studies where cells are taken from the patient and treated in a dish with a gene therapy reagent and then delivered back intravenously or to the target organ with these to treat these other diseases. So it's been so exciting to see the field develop. Gene therapy is now being tested in all of these different retinal diseases, which include Stargardt disease, different forms of congenital blindness, LCA, X-linked retinoschisis, peroxisomal storage disease, it was using gyrate atrophy in their plans going forward for gyrate atrophy, and some terrible neurodegenerative diseases like Batten disease. And further, it's now being used in diseases which are much more common. Age-related macular degeneration, for one, and in particular, neovascular form of age-related macular degeneration. Further, there are multiple strategies being used. The technology is so exciting and keeps developing. We did what's called gene augmentation, delivering a normal copy of the gene, but now there are ways of doing gene editing using what's called CRISPR, delivering reagents which could be used theoretically in any form of genetic form of blindness to support the remaining cells of the retina, so-called neurotrophic or metabolic support therapy. Optogenetics, which is so science fiction, is being done here at CIRA, where the cells have degenerated in the retina and the remaining cells are rendered light sensitive, and which is really quite extraordinary. And there are different routes of delivery being tested. So I think the prospects for gene therapy are great. I think the technology is growing quickly. As I mentioned, there are multiple strategies that are under study, and the vectors, the gene delivery reagents, continue to be improved and developed. And there's a lot more experience. Gene therapy is now in hospitals. It's now in pharmacies. It's now billed by insurance. There are now cell models that are used to get proof of concepts. You don't necessarily need to use an animal model. You can test your reagent in a dish and then make sure it's safe in a large animal model before you deliver it to human. But that will surely expedite the progress. And so it's very exciting. But there are challenges, and one of the huge ones is funding. Particularly now, since it's been, especially in the United States, huge amounts of inflation. Investors are worried about especially about clinical trials which enroll or develop a therapy that's only that would treat disease in only a small population of people. They're not so concerned about very large populations, but a lot of these inherited retinal diseases affect small pockets of people. Together, they affect huge numbers of people, but this is a real concern. And so we're really grateful to the patients and the organizations such as CIRA and the patient advocate and foundations for pushing forward this and keeping the excitement and the progress moving. There are other things which I think will develop. I think there is now prospects for office delivery instead of using general anesthesia to put somebody to sleep and do a subretinal injection the way I showed. There are ways of doing that. People are working, including here at CIRA, ways of regulating the expression levels of the gene, because you don't want too little, you don't want too much, you want to keep it, you want to have it be able to modulatable if the disease waxes and wanes. We need to get more outcome measures because these different diseases cause different disease symptoms. And we need to continue to educate the regulators and the population and continue to get natural history studies and tell the patients, especially tell the patients that there's hope, that they can help contribute to the development of treatments for these devastating diseases by testing themselves for the genetic cause of their disease, by enrolling in natural history studies to help figure out how to measure progress in these diseases, et cetera. And my prediction is that the progress will go along a curve like this, which shows the progress of development of monoclonal antibodies, which are now actually being delivered by gene therapy. Treatment for neovascular age-related macular degeneration is now using a similar process. This shows the use of cell models, and CIRA is a master at this. You can actually take cells from a person who's affected with a disease, de-differentiate them in a dish, and then grow them along a cell-specific lineage to grow your own retinas in a dish, or specific cell layers of the retina in a dish, and test treatments there. The imaging has gotten amazing, so you can look at rates of progression of the disease, different outcome measures. This shows light sensitivity, which is a wonderful one. And we've been working with virtual reality tests of functional vision. This actually shows results of one of our patients in an LCA5 gene therapy clinical trial, where in the base, you can see baseline. This is colorized and cartoon made to see. This person is looking around for objects. What he's supposed to be doing is finding them, tapping them, and making them disappear. And here, he's doing it, no problem, after his treatment. Whereas beforehand, he's just guessing. He's looking around. So we're excited about, I never thought I would be using entertainment stuff like virtual reality to apply to these conditions. But it's a really interesting time. So to summarize, it took a long time to develop the first proof of concept to go from proof of concept to FDA approval, but I think things will speed up now, and there's so many trials that are very exciting, that are either in process or being planned, and we hope that that this will create a path for developing treatments for many of the diseases for which we don't have any treatment right now. So I'd like to, first of all, acknowledge all of the funding agencies and particularly the patients who led the way, and the dogs who led the way as well, and to thank all of you for your attention. Take care.
Well, Jean, that was absolutely superb, inspirational, so much vision in all senses of the words in that presentation. The vision to start this program in the first place, the vision to see it through, and the vision restored at the end. Awesome work. I'm sure you're going to have questions for Jean, and so what I thought we'd do, we've got a little bit of time left. Jean, if you'd like to join us on the stage here in the nearest chair to me. And there will be questions, I'm sure, about what Jean's doing, maybe some questions about how that overlaps with what some of our researchers are doing at CIRA. And I also know that if you're not a scientist or an eye doctor, we sometimes talk in riddles, don't we? We use too much jargon sometimes. Jean doesn't. She's the example of how to do this right. But I think it's important that we use language that makes sense, and so to keep us on track tonight and not get too sciencey in what we're doing, we've asked CIRA's Consumer Involvement and Advocacy Lead, that's Kelly Schultz, to join us. So Kelly, if you'd like to come up to the stage also. So Kelly leads CIRA's new consumer program, and before joining CIRA, she held senior corporate roles in complaints at Telstra. You can imagine that was a busy job. Accessibility, customer experience, brands and communications. And she also holds board roles in disability service provision. She's a director of the Victorian Disability Worker Registration Board and a member of Swinburne University's Technology Innovation and Value Creation Committee. And Kelly described herself to me when she first met me as blind, but with just enough vision to be dangerous, which I thought was a great description. So anybody who's worked with her knows that she will have no hesitation in posing questions that others are too scared to ask, and she'll be ably assisted by her guide dog, Velvet, who's a key presence around CIRA as well. So welcome to Kelly. So just before I take a seat and join the panel, I'd like to point out that we've got Matt and Gink in the audience who have some microphones, and if you're watching online, Callum is moderating the questions. So please do ask us some questions. Thank you.
Thanks, Keith. Well, firstly, I'd like to express my gratitude to Jean. I am disappointed that this whole talk wasn't called Gene Therapy. But I bet that's not the first time anyone said that to you, so thank you for indulging me with that. And because they gave me the microphone, I get to ask self-interested questions in the first instance, so thank you for that. Rare diseases, you mentioned that funding is one of the biggest issues, and also funding for rare conditions. What's your top tip, and I'll ask both of you this, top tip for rallying people to care about rare diseases, be that funders, anyone else, researchers? How do you get them to care?
I feel that it's very important to speak about the work, and I, you know, like early on when we saw that we could make blind puppies see, I thought that was a eureka moment. We could use that because it's so understandable to the layman about how important it is to translate the research findings to test that sort of intervention in humans. My colleagues thought that was appalling. They were really scared that there would be animal rapists firebombing us and doing terrible things. But it had the desired effect. People got very excited about that. In fact, our first gene therapy treated dogs were on Good Morning America. And I think it led to a huge amount of enthusiasm. I'm a firm believer in educating people about what is possible. And I think it's really important just to share the hope.
I absolutely agree. And I think the other thing that we often point out is, yes, individually these diseases are rare, but when you group these inherited retinal diseases together, you've got the leading cause of blindness in the working age population. And it will take a long time to do these diseases one at a time. And so one of the things that we're working on at CIRA is how do we develop therapies that work for more than one individual condition, so that we call them disease agnostic. In other words, they don't care so much about what the disease is, but they still have an effect. And I think that's one of the important ways that we can do this. But for the individual rare diseases, I think it's really important to, you know, you demonstrated so beautifully the effective advocacy and how it made a difference in some of these conditions, and you had the naysayers, and it's the families and the kids and the demonstration of what this actually has in terms of impact. And I think the FDA are right. I mean, who cares about the pupil? You're wanting to see the kids running around the room.
Kids and puppies, that's a winner. Kids and puppies. All right, there's bound to be lots of questions out there, so I'll throw to Genk and Matt. Who's got one?
Hi, Jean. Your talk was absolutely stellar. I wanted to ask, you know, you as a scientist, you as a clinician, not everything goes right during your journey. There are so many obstacles. When you hit all those obstacles through the 30 years of discovery of this amazing treatment, what was it that kept you going, that kept you getting up and going, no, this is going to work?
There were so many times when it was just so frustrating. An obstacle would be thrown our way, which, and very unpredictable. So it's too many to count. And what kept me going was I knew this was going to be possible. I knew it would work. And or at least I wanted to get the chance to test whether it would work. And so I kept basically putting one foot ahead of the other, thinking, okay, if I hit a really firm dead like block, then I'll just give up. But I just stuck my teeth in it and wouldn't say no.
Thank you, Ingmar Wahlquist. Jean, you touched on the duration of effect and redosing, and I just wondered where we are actually on the duration of effect of these gene therapies, how we're going on redosing, possibly multiple times, and in that context also, where you think what are the best bets in terms of newer delivery vectors for these therapies?
That's a key question. We are now close to the 10-year mark for the individuals treated in the registrational trial for Luxturna, and it will be really important to see how well that's done, whether there is durability in the effect. We've had a challenge. The whole world has had a challenge in terms of the pandemic lockdown. And many of those people have not come back, or were awaiting their return to come back, because they're from around the world. There are a lot of them from Europe, one from India, they're from all over the United States. So I think we need numbers to be able to get good statistical view of how does this work. But from the numbers I've seen, the people have maintained benefits that are way over baseline. Some of them may be going a little bit down. But I think 10 years is a pretty good amount of time, because our vision deteriorates as we age a little bit too. So, it may be acceptable. If it may be possible to redose the retina if the effect wanes, and we've done experiments on that in animal models to see if it's possible, and it does appear to be safe and possible, but that has not been tested in humans, and it will be another big step to test that. I don't know that there's any plan to test it, but yeah, good question. Time will tell.
Time is definitely a big one in research. Keith, 10 years ago, if they were trialling Luxturna, what are we doing right now at CIRA that's going to be your croc lecture in 10 years' time?
Well, that's a big question. I had my chance last year. I'd probably take another 10 years to build up that amount of work. So I mean, I think there's some amazing things going on, and what we're trying to work towards at CIRA is gene therapy 2.0. And the reason is that there've been fantastic success with Luxturna, and there are other candidates coming along, but these are chosen because they are the more doable with the current technology that we have now. So for example, the AAV, the adeno-associated virus that Jean showed us, is a fantastic vector, but it's got one big issue, and that's it can only hold a certain amount of information. So it's like you being limited by the size of your text message. Remember in the days when you only had so many characters and you had to be really efficient with how you did it? Well, it's a bit like that with AAV, and if you've got a long message you need to send, it won't work for you, and you've either got to use multiple viruses and target the same cell, or you've got to find a bigger message capacity. So we at CIRA are looking to, and one of the other, so there's new vectors that can hold more cargo for some of the bigger genes that we have. That's a big one, I think, that we're looking for. And then what we want to be able to do is control the gene therapy. At the moment, when we put the gene in, we've got then no control, and that may be okay if you're replacing a missing gene, but when you're giving a gene which has protective effects, or you're using gene therapy in a different way, it's possible to overdo it potentially, and so you want to be able to turn it on or turn it off so you can tune it, and I think that's going to be a big thing. And thirdly, it was around the disease agnostic treatments. So we're setting our researchers a challenge to come up with therapies that work for many different types of disease. And they may not cure that disease, but if they can buy 10 years, while other therapies are developed, that's still hugely important. So those are some of the things that we're trying to do.
I'm looking forward to it. Callum, have we got any online questions?
We do. A viewer online has asked if I would like to how do I become part of a trial?
Well, the trials have very specific recruitment criteria for different types of condition and disease. We have a part of the website at CIRA, so we have an ability, a portion of the website where you can actually register yourself as interested in clinical trials and give us a little bit of baseline information, and then we can contact you to acquire more information. I think for inherited retinal diseases, we have a very mature natural history study which is running, and we're really interested in people who we're not already in touch with who have inherited retinal disease, and so we'd be particularly keen to have approaches from people or families with inherited retinal disease that aren't already in contact with us. But the website is full of information. I think, and we have hundreds and hundreds of people, more than 1,000 people now, I think, that have registered their interest in taking part in clinical trials through the website. So that's a great place to start.
CIRA.org.au, in case anyone was wondering. I'm sure there are more questions in the room.
Can I please ask a question? This question's to Jean. So, at CIRA, we're investing in all sorts of gene therapies at the moment. My question to you would be, if there's an eye disease that's caused by multiple different genes, how do you anticipate gene therapy would work for that particular eye disease caused by multiple genes?
Oh, that's a challenging one. I have not encountered that, but theoretically, let's say there are multiple genes that cause an autosomal disease, recessive disease, where there's probably lack of function of these different genes. You could deliver multiple vectors to the same cell, which contain the different corrective genes. Or you could take an approach where you do a gene agnostic approach that you just heard about, where you would give a treatment that would be irrespective of the genetic cause of the disease.
And I think that's what we're doing for diseases like glaucoma, where glaucoma is only a small proportion of glaucoma is caused by a single missing gene. So there is a small proportion, and it's often the early onset types of glaucoma or the very strongly running in families type of glaucoma. The rest isn't, and it's a polygenetic disease, and your risk it's due to a blend of different genetic risk factors. But in that situation, we're doing exactly that. So we're using neurotrophic factors. Those are factors that nourish the particular cells, in my case, glaucoma, and make them more resistant to damage. So glaucoma patients lose these cells faster. If we can toughen them up and make them more resistant to injury, that can have a beneficial effect. So that's one way you can do it.
Thank you. That's really helpful.
I've heard you both talk about missing gene and defective genes. Can we do both with gene therapy?
I'm sorry, missing gene?
Missing gene or defective genes. They can both be replaced.
Yes.
Amazing. Question?
Yeah, I think we have a question in front.
One of the issues with gene therapy at the moment is the cost. And certainly the incidence of various genetic diseases varies across the world. And some of the areas of the world that have a higher rate are actually poorer areas of the world. How do we see that we're going to be able to deliver gene therapy to those poorer areas of the world?
That is a very real problem. In fact, the cost of the gene therapies that have been approved so far is staggering. The Luxturna itself in the United States is $850,000 to have both eyes injected, and not always covered by insurance. And the other gene therapies that have been approved are even more expensive. I believe that the one for spinal muscular atrophy is something like $2 million, and the one for hemophilia is $2.5 million. And that's going to be a really big problem. We're having trouble, at least in the United States, along with the cost of drugs and insurance. And my hope is that as these drugs, these special drugs are generated, that the cost will go down. There will be competition. The ways of manufacturing them will be streamlined. I think a lot of the companies are trying to charge to help fuel their research pipelines, and hopefully that will be reined in a bit without hampering their progress.
I'll say one word, which is technology. So the progress in terms of the cost of new technology is incredible. And you just have to look at the development in chips. You know, you've got so much more power in your mobile phone now than you took the first, you know, people to the moon. If you look at how much it costs to sequence the first human genome, we're talking billions and billions of dollars. It can now be done for less than $100 a time. But in terms of gene therapy, you say, well, that wouldn't ever apply to gene therapy. You know, you're never going to have a mass campaign where you're going to take, you know, some sort of nanoparticle which has genetic material into it and give it to millions of people, and it will change gene expression such that protein expression changes in cell for a therapeutic effect. And of course, if you'd said to anybody, you know, we're going to be using mRNA therapies in 2020, in the early part of 2020, you'd have said, well, we're crazy. That's the decade away. 4 years later, we have delivered almost 19 billion doses of this to countries all around the world using this technology that we didn't even know how to do 4 years ago. It's incredible how fast things can change. You know, and so I would just use that as an example. It's sort of gene therapy. It's sort of taking something, a particle with a gene in it, and using it therapeutically. Yes, making AAV to clinical grade is expensive currently. So are so many other things that we have had in the past. And new manufacturing techniques, new techniques of making these viruses, new non-viral vectors that are cheaper and easier to make, and advances in technology, I think, will solve this problem, as it has for the production of antibodies, which are now absolutely routine part of clinical care, whereas they were seen as only, you know, and they had the same cost issues at the start of the antibody revolution as well. So I'm optimistic.
One more question. We've got time for one more question before we wrap up.
Thank you, Jean and Keith and Kelly. I'm really interested, following up on your point, Keith, about how quickly things are changing. What are your thoughts about the opportunities and the challenges presented by AI in this field?
Thanks, Jean.
Maybe you'd like to.
So I think AI is being used across medicine, and one of the big areas now is in terms of drug development. And so one of the things that's really important for drug development is the physical structure of proteins and the physical structure, well, particularly the physical structure of proteins. And in the past, determining the physical structure of proteins has been extremely labor-intensive. You'd put a PhD student on it for 3 years, and if you're lucky at the end of that, they'd have some sort of idea of the structure. AI approaches are now doing this at incredible speed, and almost to the extent that all protein structure will be known, you know, from the sequence, and that will be done by AI. So, and you know, so in terms of target development, I think AI is a really important technology, and a lot of what we do is try to identify targets for new therapies, as well as designing those therapies. You're trying to make the target, you know, make the key fit the lock, as it were, and so you're currently looking for new technology that can help you predict how to design your key to fit a particular lock, and I think that will be a really important thing that AI does. I think another thing it will help us, you know, find patients for clinical trials as well, because, and that's something that's building, because AI interpretation of images, you know, we spend years learning how to detect these and these different phenotypes by looking at the retina, and we may have the genetic structure, but it's still important to look at the retina, and AI can learn how to do that part for us so that it identifies disease more effectively. That's just 2 examples, but there are many more.
Thank you, everybody. Thank you for the questions. I'm sure there are many more. Now, because Jean and Keith have done all the work tonight, I'm going to ask them both to give us their call to action. So these people haven't done anything yet. What is the one thing they need to do once we're finished here? What is the call to action? What's the one thing we need these people to do for us?
I think you need to spread word that there's hope for developing treatments for forms of blindness for which we have none at this point of time.
I couldn't put it better, and I have to leave the last word to Jean. That's it. Okay, well, thanks. We're getting towards the end now. So again, if there's anything we didn't get to or you've got an area of interest, you can contact us via the website as well, which is cira.org.au. It's community@cira.org.au for the email. So I'd just like to formally thank Jean again for a phenomenal lecture. I hope you will agree that it was truly inspirational, and can see why we wanted to bring Jean all the way from Philadelphia. I don't think it was kicking and screaming, because she's been exploring Tasmania, she's been hiking, she's been getting to see a bit of New South Wales, and she's loving Melbourne most of all. So there you go. So just to conclude, I'd like to invite Jean to join me here at the lectern, and for Gerard Croc's daughter, Marnie O'Brien, Dr. Marnie O'Brien, to join me in presenting a small token of our appreciation to Jean. So we'll come around to get some pictures.
Jean, thank you. That was absolutely extraordinary. Thank you so much. It's wonderful. Congratulations.
Thank you so much.
Thank you.
Do you want to come around to here to take a picture? Yeah, just get a better shot from there. Come first. I've done enough. So thanks again to everyone for coming tonight. Thanks to all who watched online. With the Centre for Eye Research Australia, are really grateful for all your support and interest. Have a safe trip home and enjoy the rest of your evening. Thank you.